So as Jonathan said, um, my name's Associate Professor, sorry, Jimmy Breen, and I'm Chief Scientist of a research group called Black Oak Data Labs, which I'll go into in a little bit. So the title of my talk was Improving Precision Healthcare for Underrepresented and Genetically Diverse Global Populations. But for the point of this particular talk, I'll be more talking about less about sort of the global populations and more talking about Indigenous Australians, makes up the majority of the work that I do. So, which is customary here in Australia, but I just want to acknowledge that I am coming to you from the lands of the Kaurna people, and I want to acknowledge the feelings of attachment and relationship to the Kaurna people have to their place, reminding everybody that this is a land that was never ceded. Also want to pay my respects to the cultural authority of Indigenous Australians, Aboriginal and Torres Strait Islanders from all around Australia, and also pay my respects to their elders, past and present, and also to acknowledge any Indigenous person who's joining me here today. So where I'm currently coming from is Adelaide in South Australia. And actually, the river that goes through Adelaide is in this beautiful Kaurna country is actually called the River Torrens, which is the more colonial name, but it's actually called Garawirapari. And Garawirapari means red gum forest river. And actually, if you see this photo on the right-hand side, you'll see that there's along the banks of this particular river, going from the hills all the way into the ocean, is these beautiful sort of gum trees, and that kind of lines all. And this is what the name of the Kaurna people called this particular place. So today I'm going to talk a little bit about the work that we do here in Australia. I'm going to talk really about the healthcare context of Indigenous Australians and sort of, why we're engaging in sort of pharmaceutical, companies and things like that. I'm going to talk primarily around the unique genetic history of Aboriginal and Torres Strait Islanders and how that impacts a lot of the work that we do in terms of genetic genome-wide association studies and precision healthcare. And I'm really going to talk about some problems and then some potential solutions, and the solutions that we're working on at the moment. The first one is more of an ethical and a cultural one, of like sort of the lack of engagement with Indigenous communities for scientific research, from researchers. And then more of a technical one. So we'll be talking about investigating novel and unexplored genetic variation in Indigenous precision healthcare, knowing that unique history that Indigenous Australians have. So as I said, John lovely said on the introduction, is that I work for an Indigenous genomics group called Black Oak Data Labs, and we're actually found in multiple places in Australia. The majority of us are here in South Australia. If you can see my cursor, this big thing in the middle is Adelaide Oval, and we're just basically, our offices are just on North Terrace there. However, the main funding group that we work for is the Kids Research Institute, which is in Perth Children's Hospital on the left-hand side there. And then we do a whole lot of work with the National Centre for Indigenous Genomics, which is in Canberra, in Australia's capital. And I work for a lot of different groups in terms of funding groups, and where we get a lot of money in to investigate and invest in precision healthcare for Indigenous Australians. So as I said, we primarily do Indigenous genomics. I'm a genomics and bioinformatics researcher for a long time, and we're looking at more public health, precision public health, and we're mainly based in Adelaide, although we have team members in Perth, Canberra, we've got some on the Gold Coast, in Tasmania, so we're all across Australia. We're focused on the multi-omics of complex diseases, and that's my primary research focus. Also, we establish sort of precision healthcare approaches that will aid diagnosis and treatment. We develop a national Indigenous genomics ecosystem through extensive collaborative network and community partnerships. We aim to identify and advance data sovereignty and governance frameworks, which I'll go a little bit into as well. And really, a primary driver of a lot of the work that I've done for a long period of time is teaching younger researchers, from sort of tertiary education about Indigenous genomics, and more importantly, teaching Indigenous people about how to work in genomics and working with their own data. So, I mean, to set the context of Indigenous Australians, there is a large gap in the efficacy of a lot of the treatments and precision health programs designed for Indigenous Australians, and there's a large mortality gap between Aboriginal and Torres Strait Islanders to non-Indigenous citizens. I forgot to mention also, I'm a non-Indigenous man working in this field, so please bear that in mind. In terms of the diseases that mainly impact Indigenous Australians, we're talking more of now about cancer, cardiovascular disease, we have chronic kidney disease, and a lot of this is driven by diabetes. And in terms of the gap, this gap has been there for a long period of time, and because of COVID, really increased in sort of the in terms of age stratified mortality. But really, Indigenous Australians have really been a core part of the Australian continent for a long, long period of time. I always like to show this slide because it kind of puts into context the unique timeline that we have. And if we're thinking about around 1600, first European contacts with the Australian mainland, really it's been very recent that we've had the pyramids at Giza and sort of cultures within South America and Europe. But really, Indigenous Australians have been here for such a long time to put into context of the sort of the unique history that they have. Going back to about 60,000 years ago, there's evidence of rock shelters in the far north of the country, where sort of paintings have been there. There was a human remains called Mungo Man that was found in 1974 that's dated to 42,000 years ago, and close by was Mungo Woman as well. But actually more closer to home in South Australia where I live, and we have two Adamanya team members in our group. But there are stories in their culture about a giant wombat called the Diprotodon that has, in terms of their Dreamtime stories and their religious stories, and that has been extinct for over 20,000 years. So, there's been this continuation of this culture for a long period of time. And when I'm talking about Indigenous Australians, I also want to make clear that when I'm talking about two separate Indigenous peoples, so there's Aboriginal and Torres Strait Islander people, which are the majority, Aboriginal Australians, which is the majority of the mainland of Australia, which has this flag on down the bottom here. And then there are Torres Strait Islander Australians, which are for the far north, this section up near the Torres Strait here as it connects to Papua New Guinea. So the first estimated arrival on the Australian continent is about 65,000 years ago, and it's described as the oldest continuous culture in the world. And because it has been so isolated and these people are still around. There is about 163 languages spoken, but there was probably about 250 before European arrival. There's some really interesting aspects of trade with across the Torres Strait and with moccasins in Indonesia for at least sort of 500 years. And then the population is about 3.2% of the Australian total population, which is about 820,000 people. This also, beyond that, we have probably about 350,000 Pacific Islanders, which also includes about 150,000 of those 350,000 are Maori people from Aotearoa, New Zealand. So these many communities each have their very, very own unique genetic history. And we know that as during very recently, there's two Nature papers that came out of our colleagues in Canberra, Hardik Patel and Azir Hermes at the National Centre for Indigenous Genomics, which really found that, looking at the global known genetic variation that is in global communities and global databases, about 1 in 5 variants were unique in the Indigenous data that was sampled. Now, of this these new diversity that was identified, about 10 to 31% of those variants are community specific, meaning that these communities, Yarrabah, Tijikala, Tiwi Islands and Galawinku, those are all about incredibly diverse between these communities. We've only assessed 4 of a large section of the Australian Indigenous communities. Each individual one will have an incredibly a large amount of diversity. One of the kind of interesting components of that, and the reason why it's really important for precision healthcare, is if we were treating someone based on the diversity between neighbouring communities, say for example someone from Western Australia as opposed to someone from Northern Territory, it would be the equivalent of treating someone with the same, in the same way as a person who's from the UK compared to someone who's from Cambodia. So that's the scale of genetic diversity that's found within these communities. In terms of the inequality across global databases, there really isn't any Indigenous Australian genome data that's out there, let alone used for precision healthcare tools development. So we have on the left-hand side, the GWAS catalogue. I think this is a little bit old now, 2019, but the overwhelming component of that is individuals from European backgrounds. And then we have a lot of East Asian now, and that's also increasing, but really there's absolutely no section there. I think we've got a Greater Middle Eastern, Native American and Oceanian, which is basically, it would encompass Indigenous Australians, and there's very little. The impact of that is that when we build things like polygenic risk scores, we have, even with multi-ancestry approaches, we have this reduction of accuracy in terms of compared to a European individual. So if we, African individuals, if we compare the predictive accuracy of those polygenic risk scores, we see a large reduction in what we can use. If we're using that in the clinic, in primary care, we're going to see less accuracy of those clinical risk scores. So number one, in terms of the problems that I kind of posed when I was talking in the introduction, was given the unique and underexplored genetic diversity and the expanding healthcare gap between Indigenous Australians and non-Indigenous Australians, why hasn't there been more genetic research carried out on Indigenous Australians? And really, it's a reasonably important sort of question to pose, but really there's a large historical context about why Indigenous Australians do not feel comfortable engaging with the scientific community. And the number one reason for that is that a lot of the exploitation of Indigenous Australians has really been historically carried out by the scientific community. These are some pictures in which we can see when colonisation happened in the 16 and 1700s. We see that there was a lot of, quite racist experiments carried out on Indigenous Australians, being that they felt that they were inferior beings and that they were somehow sort of less human than the Europeans that were colonising the area. And really, a lot of Indigenous Australians were shunted away, taken away from their Indigenous lands, put onto communities, and also treated incredibly badly. A lot of that treatment still lingers today, but there's also been this sort of more engagement in trying to improve the situation. So in terms of like I said, the answers to that question would be the limited engagement within, research projects, exploitation of participants' information, as well as their intellectual property, and majorly with limited benefit to the communities that were actually studied. And there really wasn't any formal plans to build capacity with Indigenous communities to really engage in some healthcare and scientific research that could potentially really transform their lives for the better. And there are lots of frameworks now that are available that we can use to really improve this situation. So I'm sure many of you are probably aware of the FAIR principles for data and the use of data. So we're talking about findable, accessible, interoperable, and reusable data to use for benefit of the scientific community. But there's also the CARE principles, which is recognising the collective benefit of the research to the communities to use. Obviously, this is all communities, not necessarily Indigenous communities. giving peoples the authority to control their own data and their rights and interests of Indigenous peoples. The responsibility, so those working with Indigenous data, the responsibility to share how those data would be used to support Indigenous people's self-determination and collective benefit, and really building an ethical framework and to respect the Indigenous people's rights and well-being, the primary concern of the research project, and the reuse of a lot of this data. So potential solutions. And this is a lot of the work that we're doing here in Australia, is developing governance structures for research projects involving Indigenous communities, and also develop meaningful research relationships based on a community benefit. So in our team in Australia, we have been sort of part of the movement to develop data access committees and Indigenous governance committees, councils, that all work within the jurisdictions of state and territories to really assess the appropriateness of each individual research project that would happen within the communities in states and territories. So these are all the people in our National Indigenous Governance Committee through the Australian Alliance for Indigenous Genomics, and we have state-based committees as well, where researchers would pose proposals, and that Indigenous people will say yes or no to the research in which they do, and then they'll engage in the benefit to the community through that group. I also am funded through a Canadian Indigenous community grant called, around marine biomass innovation, and that's in the island of Newfoundland in Canada. And through my postdoc, Adam Hederick, as you can see on the right-hand side there, we've developed the same data access committees in that particular project, interacting with the Mi'kmaq people of Western Newfoundland. In terms of here in Australia, and as you probably heard from Jonathan, in terms of the gap between Indigenous Australians, we engaged in what's called the prophecy study, and the guy on the right-hand bottom right there is our director, Professor Alex Brown. And in 2016, they went to communities and said, okay, we want to do a large cohort study to really improve the benefit of the lives of Indigenous Australians. And what is the research question that we should focus on? And overwhelmingly, the largest question that everybody asked was about type 2 diabetes, and that being a primary driver of a lot of the problems associated within community in terms of their healthcare. So in terms of Indigenous Australians, they see a greater burden of disease in their communities in South Australia. They see an early onset phenotype of a lot of diabetes. They see greater complications such as diabetic retinopathy, kidney disease, cardiovascular disease, and cognitive disabilities. And also we're seeing the start of a lot of intergenerational impacts of diabetes across to the next generation, something that we want to avoid and stop in the future. So the prophecy study, which is colloquially called the Aboriginal Diabetes Study within community, was designed to unlock and predict and prevent diabetes and related disorders. Actually, funnily enough, the number one question that a lot of people had was, why do two people who have got type 2 diabetes, why does one person get an additional large complication? For example, they get really bad kidney disease and they have to be on dialysis, while another person with type 2 diabetes can live healthily or reasonably healthily for the rest of their lives on just basic treatment. So in this particular prophecy cohort, we've sequenced around 1,250 Indigenous South Australians. We've assessed their multiome, so we've got whole genome sequencing, epigenome, full transcriptomes, proteome, metabolome, lipidomes of every individual. And then we're currently following up with wave 2 assessments, which should be finished at the end of next year. And then we'll potentially do a wave 3 follow-up, helping us to design predictive measurements of type 2 diabetes and related disorders. We also have this incredibly large environmental questionnaires and metadata that can be helped to build into these models. So Moving towards more of a technical component, and I realise that we've got more of a technical audience today. The second question that I posed was the expansive genetic diversity that we found in Aboriginal and Torres Strait Islander genomes impacts the ability to analyse and interpret meaningful signal for us to develop precision healthcare approaches and also drug development later on in the future. So in my group, and this is mainly led by Stevie Pedersen, who's in my team, he's on the top right-hand side there. They've done an incredibly large amount of R package development over the course of the last sort of 10 years in working within multi-omics data sets that really pose and address specific questions. Steve is actually on the Bioconductor Community Development Board, it's definitely they are definitely a person that would be great to talk to in terms of developing a lot of the R packages in the future. So one of the ones I wanted to focus in on is this transmog, which is a package to create new reference genomes and reference transcriptome using a set of variants. Now, why would we want to develop a package like this? Well, in terms of diverse genomes, and obviously I've got Indigenous Australians, which are incredibly diverse. The most diverse genomes in global databases are African genomes. As you can see here from a study, I think out of the Salzburg's lab, which this was actually published in Nature, is that they did a African pangenome where they sequenced a lot of different African genomes and found that the genome itself, the African pangenome, so the conglomeration of all those genomes together, represents about 10% more DNA than the current reference genome actually has in it. And as you can see here, a lot of these lines on the karyotype here are bits of the genome that are undiscovered and unknown in Africans that are just completely absent in the human reference genome, GRCH38. Now, small and large genetic variants really impact reference genome alignment, and we see that in our particular work when 1 in every 5 variants is actually not known and not found in global databases. It becomes very hard to align DNA segments onto the genome. And that standard reference genome is biased towards populations from the global north. So it's impacting our ability to align DNA to the genome in order to analyze. So this is what we call reference biased, and it's particularly pronounced in multi-omic sequencing approaches where you have a slightly less representation of the genome. So for example, RNA-seq, which is a lot of the work that we do here in Adelaide, and DNA methylation, which is my colleague in Perth, Sam Buckberry does, which we're using EM-seq. These sequencing approaches do have a larger reference genome bias. So a potential solution to this would be to augment the current reference genome to be closer to your targeted population, or potentially build diversity into a global reference genome sequence. So the way that Stevie did with this particular package, Transmogar, was to augment the human reference genome to be closer to the target population. So what we've done here in Prophecy is the reference genomes on the top right, which is the faster sequence, and then Prophecy individuals through there. As you can see in these boxes, there's a particular variant that would either be rare or different than the reference sequence. And we can actually create a new reference genome or transcriptome to basically take the major allele found within the cohort if it is different from the reference genome. So Transmogar, you can take a variant file, a VCF file containing small SNPs and indels, and enables the user to modify and create a reference genome or transcriptome for analysis, which really does increase the number of sequence reads that we can use for analysis and potentially a more accurate assessment of gene expression, DNA methylation, or just sequence coverage in general. But what about larger variants? How we're talking about SNPs and small indels here, but if we can't easily add large variants to a reference genome, it's going to change our coordinates and make it very difficult for us to analyze. So going beyond this, we're also looking at other ways to be able to include such things. So as you can see here with the linear genome reference, we see sort of a large, coverage dropout in the middle here where a structural variant was increased, an insertion was added to the genome. And you can use these pangenome or genome graphs to really represent these additions or deletions into the genome reference. And so we're aiming to sort of extending this transmogar approach to individuals with global populations to really create what we describe as a personal genome. So it's a genome of an individual that has all of the variants of that individual and then compare it to global populations and reference genomes. So this will ultimately remove reference bias, but primarily can do mostly with long read sequencing, where you can enable the alignment of a complete individual haplotype. As you can see on the right-hand side here with these sort of tube map diagrams, it kind of enables everybody to be able to include all diversity found in global populations into one reference genome to remove the kind of need for more augmented types to remove reference genome bias. Another solution, and this is actually probably my research interests at the moment, is really using ancestry agnostic approaches to functionally interpret novel genetic variation using genome language models. Now, this is a really important factor for a lot of sort of drug development and a lot of genome-wide association study work, because if you can't identify the functional importance of a particular variant, then you don't have a way to include it into sort of drug development and things like that. So my group is more, so this is all the people in Black Ochre Data Labs that work with a lot of the data. We're interested in genome language models because genomes of Indigenous individuals are highly divergent from the human reference genome. Interpreting each of those unique genetic variants is difficult, especially if there's a lot of them, which there is, and they are in non-coding regions, which are difficult to interpret at the best of times. Most functional genomics data is also tissue specific and assayed from populations from the global north, mostly European and American. So to enable the development of targeted treatments for type 2 diabetes, cardiovascular disease, and other complex disease, we need sort of ancestry agnostic tools for a functional interpretation, whether it's a person from Indigenous Australian communities, East Asian, European. We need something that's universal that can work and we can use to interpret all genomes. So if I pose a target question, so given I have a non-coding or structural variant that we've identified in our work that's significantly associated with a trait, say for example, chronic kidney disease, can I predict the gene that is being impacted? Can I predict what tissue the impacted gene is likely to be active in? Can I predict the impact of the variant from a vastly different reference genome? In fact, a personalized reference of the individual. So in other words, what is the context of what this unique variant is sitting in? And with this, we've been really testing and doing a lot of work recently in using these deep learning models for assessing gene regulation. So a lot of this is built on epigenomics information from ENCODE, GTEx, things like that, on RNA-seq, histone and ChIP-seq, and DNA methylation data. And the models aim to predict the output of a regulatory assay and help identify regulatory elements within coding and non-coding areas. I kind of tend to call these the dog models. This is, a lot of the models came out of David Kelly's lab at Calico, and more recently, as you can see, dog models because we have Borzoi, Bassett, Basenji, Akita, and Saluki. And a lot of these extend a lot of the convolutional neural network models to transformers for self-attention models. And if we, in terms of transformers, I'm sure you're thinking of ChatGPT and things like that. So a lot of this came out of this paper, this archive paper, which is very famous now. I think it's the most cited paper in modern publications, where it's using self-attention to provide context to whether it be text and text mining, but in our ways it gives us the ability to substantially improve gene expression prediction accuracy from DNA sequences, and able to integrate information into long-range interactions. So the variant that we identify that's in a non-coding area, where is it actually impacting, and what are the regulatory elements that are at play? So apologies for the more fuzzy sort of figure here, but took this from sort of David Kelly's YouTube video, which you should all see. The input to a lot of these models is a input DNA sequence, whether it's a reference genome with the variant augmented in it, or whether it will be a completely native haplotype. And you put it into this particular model, and it outputs genome tracks of predicting the DNA's hypersensitivity regions, or attack-seq, or chip-seq, or anything like that you can use in your interpretation of your work. So to extend that, they've really done this this informer model, which you can see you can add here and a particular variant at a particular site, and then you can see, using the embeddings of the model, what has been impacted, what is the most important part in a 200 kb sequence around that particular variant that is impacting it's important within the model. And you can get a lot of these embeddings out. You can also use a lot of other ways, such as the gradient input, to provide a kind of a tissue and cell type specific context to that in respect to a different output. So obviously this also, you can use this with cage sequence, 5' cap sequencing, to have a look at which promoters and things are being expressed or deleted in the particular area. So these attention weights are very important for giving sort of prioritizing a particular variant and which gene or which regulatory element that are impacting, and also allows you to do a lot of discovery work with it. In us, we're really interested in sort of what the impact of a lot of the common variants and a lot of the variants are having on our transcriptomic data that we produced across the prophecy cohort. So if we can take a prophecy individual, we take whole blood, and we're able to predict the coverage of RNA sequencing or gene expression for that particular prophecy individual. We can then include a variant at a specific point and actually have a look at how it sort of compares to a lot of the eQTL work that we have through GTEx and things like that. You can actually put e-values and different things with variants at different places and have a look at how expression changes with that, whether it be a reference or alternate allele. So through all that, as you can see, we've got sort of a more ethical and technical component. Just want to also close just to say there's still significant cultural, ethical, and technical barriers to overcome in order to achieve equitable precision healthcare in Australia. And a lot of the work that we're doing really extends on, we're doing a lot of gene discovery work, but we really want this to be, a pathway to engage in pharmaceutical companies and drug development people in terms of being able to accurately assess this genetic variation, see where it impacts and is associated with a particular disease, and then potentially inform a lot of the drug treatments that we're going to be using for preventative healthcare. So engaging in this meaningful scientific and cultural exchange will ensure that everybody benefits from the research, not just the researchers, which has been the primary driver in historical contexts. So large language models have a really powerful role to play in the interpretation of novel genomic variation in underrepresented populations. And obviously also we are aware of a lot of the security issues in putting a lot of Indigenous data up online and within these models. Removing that agency that individuals have with it. So we're looking at doing local models, or also engaging in Indigenous communities and Indigenous people about how they would like us to use these models and for their work, because also bringing their extensive cultural context built from 65,000 years being on the Australian continent. And just to reiterate, this is not really just an Australian initiative. Everyone will be a benefit from assessing novel genetic variation. What we're after is not drugs that are good for an Indigenous Australian, what we're really looking for is a drug that's universally gives universal efficacy for the treatment in which they're trying to do. So we're looking for that universality of a lot of the drug treatments that we're doing. So just like to thank my team, all of the people in Black Ochre Data Labs, National Centre for Indigenous Genomics, all of the funders, NTB Connect, Heart Foundation, Canadian groups, and National Centre for National Health and Medical Research Council, sorry. And thank you for all here today, and I'm more than happy to take some questions.
Thank you, Jimmy. That was fantastic. We don't have any questions in the Q&A, so I might ask one. If a team is going through target evaluation and they have say, a gene that they are working with and they want to know how that might be affected by an Indigenous population or another subgroup, how might they go about either submitting or collaborating or evaluating whether that would work better or worse in terms of their target?
Yeah. It is, so in terms of in Australia and a lot of people, you can actually engage with, say for example, here in South Australia, we have the South Australian Aboriginal Governance Committee. And in terms of prophecy, we have sort of a research team, and they basically can assess sort of the relevance of the work. You can actually propose what's we call a sub-study, and there's a sub-study form that people can use to engage in the particular work. But ultimately, the number one thing advice I would say is you need to basically put in the effort to actually talk to communities about that particular research first. It is kind of, I realize it's hard for international, people in terms of coming and sort of starting that conversation. But really, Indigenous people are really interested in a lot of the research that happens and that has really benefited a lot of different populations around the world. And even myself, I mean, I'm also non-Indigenous, but I, through this Canadian research project, which I mentioned, I actually go to Newfoundland every year, which if you know where Adelaide is and Newfoundland is, it's on the exact opposite side of the earth. So every year I have to engage with those communities about sort of the research projects in terms of data access committees and portals and things like that. So I think people can do it. However, it does require investment, and it does require investment in time and money. Yeah.
We do have a question from the chat. Are there Indigenous researchers who serve as a kind of research champion within the communities?
Yeah, so I mean, I mentioned Professor Alex Brown. He's actually a Yuin man. He's probably one of the more sort of well-known Australian Aboriginal genomics researchers and sort of public healthcare sort of clinician in Australia. And he's certainly been the person driving a significant amount of the sort of promotion of this research in Australia. But we do see that there's quite a few different genomics, Indigenous genomics researchers in Australia that we're engaging with and have brought into the Australian Alliance for Indigenous Genomics. Not only that, Jonathan, there's also actually been a really great push for Indigenous communities around the world. So, banding together because to sort of, to use equitable genome sequencing sort of approaches. So talking Indigenous, Native Americans and Canadians, Native Hawaiians. We have sort of groups in sort of Northern Europe, Indigenous communities, Inuit populations that we engage a little bit with through the University of Copenhagen. All of these people are sort of real champions of a lot of, genomics-based healthcare, which is great. Yeah.
And I guess that leads into the other question here. Are there concerted global efforts to increase the diversity of reference genomes?
Yeah, I mean, a lot of it in terms of a more structural way, there's the Human Pangenome Reference project, which I believe is, as I sort of mentioned, those genome graphs in there. Though that was a sort of one where individual communities around the world have been kind of pushing sort of creating of reference genomes that they can use within their community. And obviously there's, like I know there's Korean, Thai, Malaysian large reference genome projects are out there that really are building sort of diversity of, the genomes in their jurisdictions. It is not necessarily done on like a far coordinated level internationally, and that's probably because it's originally was driven by a lot of Americans and sort of funding through that. And I know that is not necessarily going to be the case for for a longer period of investment. But there is a bit of a global community that can do this, as well as I mentioned, in terms of Indigenous people wanting to do it on their own terms as well. There's also the tricky component of it is actually sharing the data, because one of those components that community benefit, if we were to share a lot of the work, and Indigenous people here in Australia were like, yes, we want to put it on global databases to really impact it. Um, how does the benefits flow back to communities, and without it being taken and sort of not necessarily exploited, but a lack of those benefits going back to the communities in which they were originally sampled.